The purpose of this introduction is to understand the workflow for RNA sequencing from RNA isolation up to data analysis. We want to understand key considerations in planning mRNA and total RNA sequencing and consider them before we start processing our samples. We will then take some time to focus on the kits specifically. Again, this is part 1 of a 3-part webinar series on RNA sequencing. Part 2 will discuss best practices, and part 3 will dive into analysis. Let's begin with an introduction to RNA sequencing. When we think about RNA, it's good to think about it in contrast to DNA. DNA is double-stranded, RNA is single-stranded. DNA shows identity as the amount of DNA in a cell is almost always constant and the message is consistent. It's saying the same thing all the time. Typically, all cells in an organism, for the most part, have the same DNA, unless there's some mutations, such as in cancer. RNA, on the other hand, is indicative of activity. It's constantly responding to its environment. It's saying different things at different times. It varies from one cell type to another, and it varies in terms of different types of RNAs. In short, DNA is typically considered to be stable, while RNA is considered to be dynamic. There are multiple different features of RNA that we can focus on. For example, we can look at size. We have microRNAs that are about 20 nucleotides long, and mRNA that can be thousands of nucleotides long. We can also take a look at the shape of the RNA and how it might fold over and form secondary structures. We can take a look at its stability and how long it's present in the cell. And we can also see how much is present by looking at individual copy numbers of transcripts within that cell. We can classify these RNA species by function. The ones that we might be most familiar with are the messenger RNA, ribosomal RNA, and transfer RNA. These are the ones involved in protein synthesis. We also have RNA species that are more focused on co- or post-transcriptional modifications or on DNA replication. These would be our small nuclear and small nucleolar RNAs. Then we have regulatory RNAs, which include microRNA, pRNA, small interfering RNA, and long non-coding RNA. Together, all of these are considered our transcriptome. Keep in mind that we might have parasitic RNA as well. We can use the differences in functions and modifications of these different RNA species to zoom into specific RNAs of interest. For example, if we want to look at total RNA sequencing, we might not want to include ribosomal RNA. Our RNA makes up a high percentage of the total amount of RNA, but it may not give us a lot of useful information for our purposes if we are not focusing on studying our RNA. So in this method, to exclude the abundant rRNA transcripts, we can use Ribozero or Ribozero Plus rRNA depletion kit, which will deplete rRNA from our RNA samples. We will then capture the remaining RNA, which will include mRNA and non-coding RNA. The kits available with this method are our TruSeq Stranded Total RNA Kit with Ribozero and the Illumina Stranded Total RNA Kit bundled with Ribozero Plus. Keep in mind that due to the workflow and our bead cleanup steps, RNA species that are less than 150 nucleotides might be lost during the library prep. So ultimately, with this method, we will be capturing total RNA longer than 150 nucleotides and exclude rRNA. Another common way to enrich for a particular type of RNA is by poly-A selection. Most messenger RNA and long non-coding RNA are polyadenylated, so we can use beads that contain poly-T nucleotides to capture these specific transcripts. If we have viral RNA within cells and they're polyadenylated, these also can be captured. The kits that we have available for this method are our TruSeq Stranded mRNA, TruSeq RNA V2, and Illumina Stranded mRNA kit. Another way to target more specific transcripts is by using sequence-specific probes that will capture a subset of the total RNA. For example, our TruSeq RNA exome kit, which used to be called TruSeq RNA Access, has probes that are specific to the exome that will capture mRNA targets. With this kit, mitochondrial targets can be captured as well. For capturing RNAs with target-specific probes, we also have TruSight RNA Pan-Cancer, TruSight RNA Fusion, and Illumina RNA Prep with Enrichment Tagmentation kits. These kits have probes specific to a subset of mRNA, so if you know a little more about what you're specifically looking for, such as cancer genes for instance, one of these kits may be suitable for your needs. When we're considering which kits to use, we want to take a step back and think about our RNA-Seq applications. What are the questions that we want to address? We can generally divide these RNA-Seq applications into two buckets, profiling and analysis. Profiling is going to be more about counting. How is gene expression changing in response to treatments or in different cell types? We can take a look at gene expression profiling or allele-specific profiling. If we're looking at mRNA, we can count the different mRNA transcripts within different cells or treatment groups. We can also do this for viral RNA. In terms of analysis, the questions we ask would be, what is the sequence and are the sequences different? So this would be looking more at the sequence itself rather than counting how many transcripts are available. This would include whole transcriptome assembly, looking at novel fusions, biomarker discovery, and novel transcript discovery, among others. We can also look at genome annotation or alternative splicing, characterize linked RNAs, study small non-coding RNAs, and even discover a novel RNA virus like SARS-CoV-2. The common RNA sequencing workflow is going to start with RNA isolation onto library preparation. And once we've QC'd our libraries, we would want to move into sequencing and subsequent data analysis. So first, let's focus on RNA isolation, which is a very important part of RNA sequencing. We're often asked in tech support, what kit should I use for my RNA isolation? While we have no official recommendation, we know that customers have successfully used the Master Pure family of kits by Lucigen. They've also used Qiagen, Ambion, Trizol, etc. The main point here is that we want good yield and high quality material. We also want to make sure that we're capturing the right species. So if we want to interrogate microRNA, for example, we want to make sure that the extraction kit we use is going to be able to capture microRNAs. So what we want from RNA isolation kits is good yield and high quality material. And as always, check the literature to make sure that we're capturing the right species. Our RNA input quality will dictate which kit can be used for our needs. Earlier, I briefly mentioned these library prep kits and their different applications. If we have high quality input RNA, we're able to use any one of these kits. What we mean by high quality is RNA with RIN or RNA integrity number greater than 8. This number applies to mammalian species, so if you're working with another species, check the literature to see what would be considered high quality RNA RIN value. Not all kits are compatible with FFPE samples. If we do have FFPE samples or samples with a RIN value less than 8, we recommend using either the TruSeq or Illumina Stranded total RNA, TruSeq RNA exome, Pan-Cancer Fusion kits, or the Illumina RNA Prep with Enrichment Tagmentation Kit. These have been developed to be well-suited with FFPE. Keep in mind that input quality is the #1 most important factor for RNA-Seq, so we definitely want to check our input quality. So how do we know if our sample has high quality or not? One thing that we recommend to determine this is to run an RNA chip on a bioanalyzer. We should be able to see clean ribosomal peaks, and it should also give us an RNA integrity number. With this information, we can determine what the RNA quality actually is. Alternatively, we can run an agarose formaldehyde gel and see if those RNA bands are intact. When considering RNA input amount, there will be different input requirements for a different kit. So how do we quantify our RNA input? What we recommend is using a fluorometric method like RiboGreen or Qubit. We've found that nanodrop is not the best method for measuring input quantity, as it tends to overestimate quantification, especially if we have not DNase treated our sample efficiently. We recommend a DNase treatment, which is often included in most RNA isolation kits. Keep in mind that DNA can affect accurate input quantification, and it could show up in our library prep downstream as well. As I mentioned earlier, the RNA input quality will dictate which kit we can use. In addition, it will also dictate how much input we would need. If we have high quality input, we can use 100 nanograms to 1 microgram for our TruSeq mRNA and TruSeq Stranded total RNA kit. 10 nanogram for enrichment kits such as TruSeq RNA Exome, Pan-Cancer and RNA Fusion, or 25 to 1,000 nanograms for our Illumina Stranded mRNA Prep Ligation kit. As you can see in this slide, our newer Illumina Stranded kits accommodate a lower input amount. If we have low quality or variable quality input, we may need to add a little more input to get even coverage and good yield from our library prep. So in this case, we would still use 100 nanogram to 1 microgram for TruSeq Stranded total RNA, and as low as 10 nanograms for Illumina Stranded total RNA. You will notice that TruSeq Stranded mRNA, RNA-V2, and Illumina Stranded mRNA are not recommended for use with low or variable quality input. If you're looking for enrichment kits, we will use more input, around 20 to 100 nanograms, depending on the level of degradation and quality for our RNA Exome, Pan-Cancer, and RNA Fusion kits, and 10 to 100 nanograms for our RNA prep with enrichment workflow. The Library Prep and Array Kit Selector tool is linked here, and this is a great tool that can assist you in determining which kit is best for your project. So, once we have our RNA extracted and we know the quantity and quality of our samples, we can go ahead with our library preparation. We first need to purify our RNA, and we can usually take one of two approaches. Either we enrich for the RNA species that we want, or we deplete the RNA species that we don't want. For example, poly-A selection, which is used with TruSeq RNA-V2 and our stranded mRNA kits, will use poly-T beads to capture RNA transcripts that are polyadenylated. We will want to make sure that our RIN is greater than 8, or we're using high-quality RNA, because if the RIN value is less than 8, this may indicate variability across samples, and it can lead to various degrees of 3' bias. This is why we want to have a high RIN value with poly-A selection. Alternatively, Ribozero or Ribozero Plus depletion uses probes against our RNA to deplete the samples of these abundant transcripts. Our stranded total RNA kits use this approach. These kits are compatible with RIN less than 8 or variable RINs across our samples because even with low RIN values, we would not expect 3' bias. This also has the benefit of capturing non-coding RNAs if these are of interest to you. Because we're capturing more RNA species, it requires twice as many reads to achieve the same coverage as a poly-A selection approach would. The other option we have is a post-library enrichment approach used in TruSeq RNA Exome, RNA Fusion, RNA pan-cancer, and Illumina RNA prepload enrichment. Again, these are compatible with samples with RIN less than 8 or FFPE samples. After we do our RNA selection or RNA depletion, we need to convert RNA to DNA. So what happens is that after poly-A selection or rRNA depletion, we will fragment and randomly prime our RNA transcripts. We will then proceed onto first strand synthesis and second strand synthesis. Our method for converting RNA to DNA provides the most quantitative and uniform coverage on the market, as described by the Broad Institute study listed here, which compared different methods of preparing cDNA for sequencing. This study shows that this method gets more uniform coverage across genes, and this can be especially important for annotation or alternative splicing. Some key QC points for our final libraries. Once we finish the prep itself, we would want to check the libraries on a bioanalyzer. We want to make sure that the size is at the appropriate range that we've targeted. And we also want to look for any unexpected peaks. We recommend quantifying using qPCR, as qubit, bioanalyzer, and nanodrop are not as accurate. qPCR is typically the best method to avoid any unexpected cluster density because it quantifies complete libraries, and these are the ones that can cluster on the flow cell. If you want to learn more about best practices for RNA library preparation, tune into part 2 of the webinar series, which focuses specifically on best practices. So now that we've QC'd our library, we are going to move on to considerations for sequencing. One question related to sequencing that we get a lot at tech support is about coverage. What coverage depth do I need for my RNA-seq analysis? We know that more coverage will give us greater confidence about the sample sequence. So let's say that the sequence in the gray bar is our reference sequence, and our blue transcripts are going to be our reads that we got from our sequencing experiment. If we had one level of coverage and we see that there is a change from A to C, we wouldn't know if that was an error in alignment or an error in sequencing. We wouldn't be confident that this is a real change in sequence. But if we sequence further and continually see this difference, then we are much more confident that this is indeed supposed to be a C at this specific position for our sample. You'll notice that in the DNA sequencing world, we talk about coverage in terms of how many X coverage is needed, 30X, 100X. For RNA sequencing, we use the absolute number of reads, and this is because RNA is dynamic, so there will be varying amounts of the transcriptome in various cells or conditions. So it's more difficult to establish a specific coverage for something that is varying, which is why we use the number of reads itself. When we look at differential expression, we can again gain confidence if we increase our number of total reads. In this graph, each dot represents the expression of one gene. The X and Y axis will be two replicates. We would expect there to be a solid line across the XY axis. But as we go to the lower region of gene expression, it tends to fan out a little bit more. If we increase our number of reads, some of these low expressors move up to the higher expressing region, and we can then better tell if something is an outlier or if it just has a lower number of reads, which is why we're seeing the variability. Because we know that genes are present in different copy numbers, it's important to get the right number of reads in order to obtain confidence in what our gene expression actually is. So more reads should result in fewer genes being in that low section. So how many reads are actually needed? If we're looking at RNA profiling or counting gene expression within the cell with our mRNA libraries, we could use around 10 million reads per sample based on a human-sized transcriptome. If we want to go a bit deeper and do a transcriptome analysis, we would need a bit more, around 25 million reads per sample. These are just general numbers. We highly recommend going to the literature to see what would be best for your species and for your specific application. These numbers are more specific for mRNA samples. If we have rRNA depleted libraries, if they're from high quality input, we'll probably need around 2 times more reads. For rRNA depleted libraries with FFPE samples, we will require a bit more, around 4 times as many reads. And part of the reason for this is that FFPE samples tend to preferentially lose exonic content. So we want to increase our read depth to get more confidence for those sequences. For our RNA enrichment, exome, pan-cancer, and fusion kits, we have a set number of reads that we recommend. For RNA enrichment with a respiratory virus oligo panel, 500,000 reads per sample. For RNA exome libraries, 25 million reads per sample. RNA pan-cancer or RNA fusion, 3 million reads per sample. Again, these are general estimates that can be used as a reference. We should always go to the ENCODE guidelines, which is a great place to start. And definitely check the literature to see what's important and recommended for your specific application. In terms of run type, we often recommend a single read for profiling-based applications and paired end reads for analysis-based applications. Paired end reads will decrease percent duplicates, increase percent alignment, and give us more uniform coverage across genes. Because our stranded mRNA and stranded total RNA kits give us stranded information, we know that read 1 has better coverage at the 3' end, and read 2 has better coverage at the 5' end. So if we go ahead and perform paired end sequencing, we'll be able to get better coverage at both ends. Paired end reads also help with novel transcript discovery and gene structure characterization. Keep in mind that we will want to ensure that our analysis software can handle paired end data. While most aligners do well with paired end data, it's always great to check. Now, what do we mean by decreasing duplicate percentage? Let's take a look at this reference genome. The exons are the boxes in gray. The blue boxes are the reads for our sequencing run. If we were to do a single read for all 3 of these transcripts, most likely we would not read through the insert. So if we did a single read, 1/75 run, it might look like we have the same start and stop site for every one of these reads. But if we go ahead and do the paired end run, we would notice that we have a different stop site for each one of these transcripts. So we will know that these are not duplicates, and they are in fact coming from unique reads. In terms of read length, our general recommendation is 2/75. This is a very general recommendation, and it will vary based on the application. So you can use this as a reference, and as always, check the literature. Now let's move on to our kit options for RNA sequencing. We have 6 mRNA library prep kit options. The differences between these kits lie in how they capture RNA, the output, and the analysis that can be performed downstream. These kits are TruSeq RNA V2, TruSeq Stranded mRNA, Illumina Stranded mRNA Prep Ligation, TruSeq RNA Exome, and Smarter Ultra Low with Nextera XT. We will start with TruSeq RNA V2, which is considered our original RNA kit, and it has been widely used for many years. We know that mRNA is polyadenylated, so we use the poly A tail on the 3' end to capture mRNA. To do so, we utilize beads that have poly T, which will bind to the poly A tail. We will then go into fragmentation and random priming. The reason that we have to fragment our material is that our sequencers cannot sequence very long pieces of sequencing library. So we have to bring it to a more appropriate size for the instrument to sequence. We will then enter first and second strand synthesis. At this point, we will have cDNA, which is essentially double-stranded DNA. We will then create blunt ends with end repair. Add an A to the 3' end during A tailing. And this A will be used to ligate the adapter onto the fragment. Keep in mind that this is a TruSeq chemistry, which means that we will utilize a ligation-based method for adding the adapters. Once the adapters have been added, we will have a sequencing-ready library. The information in this slide is divided into 3 parts: input on the left, protocol in the middle, and output on the right. And I will keep this formatting the same throughout the presentation. So for TruSeq RNA V2, we can use 2 input types. If we use total RNA, we can use 100 nanograms to 1 microgram. We will want to use high-quality material from eukaryotes. And the reason that we want high-quality material is because we're using the poly A tail located on the 3' end for the pull down. If we were to use degraded material, it could be fragmented or shorter in length, so we could have 3' bias because we would only see the area on the 3' end near the poly A tail, and not the areas in the middle and the 5' end. By having high-quality material, we will be able to pull down a longer intact mRNA. The second type of input that can be used is previously isolated mRNA. If using isolated mRNA, you can use 10 to 400 nanograms of high-quality input, but you will skip the poly A selection step. This is a non-directional workflow, which means that if knowing sense versus anti-sense information is important to you, this may not be the kit to use. You can multiplex up to 24 samples, and it requires around 4.5 hours of hands-on time. Our output here is specific for poly A selected RNA and mRNA. Now, what if we want strand information? For this, the TruSeq Stranded mRNA kit may be an option. This workflow is very similar to TruSeq RNA V2, so I'm going to only highlight the areas that are different. So we will start with our poly A selection, fragment, prime, and go into first strand cDNA synthesis. Now, the difference comes in at the second strand synthesis step. At this step, we will incorporate DUTP instead of DNTP. The incorporation of DUTP in second strand synthesis quenches the second strand during amplification because we use a DNA polymerase that will not incorporate past the U from DUTPs. This way, we will know that the material which is being amplified is from the top strand. The input and output are the same as with RNA V2. What is different is that this workflow is directional due to the incorporation of DUTP. Indexing options for this kit include 24 single indexes, 96 combinatorial dual indexes, and 96 unique dual indexes. Now, what if TruSeq Stranded mRNA sounds great, but you just don't have that much input? Then our newer Illumina Stranded mRNA prep may be a consideration. The workflow is very similar to TruSeq Stranded mRNA with key differences. First, it can accommodate a lower input, and it utilizes RNA index anchors to add indexes to the insert, and this enables multiplexing up to 384 samples. Strandedness is introduced at the cDNA second strand synthesis step, just as in the TruSeq Stranded mRNA kit, where DUTPs are incorporated into the second strand. Since the polymerase cannot incorporate past DUTP, the second strand is essentially quenched during the amplification step. The Illumina Stranded mRNA workflow requires an RNA anchor intermediate, which introduces a T overhang. This anchor enables compatibility with up to 384 unique dual indexes. The anchor itself is a Nextera sequence, identical to tagmentation adapters, plus a T for ligation compatibility. This T overhang is common to all inserts and will create a low diversity cycle during sequencing. This can lead to a random base call at this cycle, so we recommend T trimming to mitigate this. And you can find information on the T trimming process in this link bulletin. As previously mentioned, Illumina Stranded mRNA accommodates a lower input amount starting at just 25 nanograms of high-quality RNA. This is used for poly-A selection, and this is a stranded kit which will support up to 384 plexity with unique dual indexes. Now, what if you want strand information, but you still don't have enough input? For this scenario, you can use the TruSeq RNA Exome Kit, which is formerly known as TruSeq RNA Access. In this workflow, we will create libraries of all RNA present. We will fragment the RNA, make cDNA, add adapters, and create full libraries using the TruSeq Stranded mRNA workflow, but skipping the poly-A selection step. We will use sequence-specific probes to bind to sequences for the whole transcriptome. We then utilize streptavidin beads to bind the probes and pull down the libraries of interest. For this workflow, we have the option of using high or low quality input. If using high quality, we can use 10 nanograms. If we're using low quality, we will have to use a bit more, around 20 to 100 nanograms, depending on the DV200. DV200 is a measure of the percent of fragments that are longer than 200 nucleotides. This kit is compatible with only human samples, as the probe sequences used are designed for the human transcriptome. Since the upstream library prep workflow is done with TruSeq Stranded mRNA, it is stranded. There is the option to multiplex up to 24 single-indexed samples, and our output is the coding transcriptome. For ultra-low input, we can use the Smarter Ultra-Low Kit by Takara in conjunction with our Nextera XC kit. In this workflow, we will use the Smarter Ultra-Low Kit to perform whole transcriptome amplification and take 1 nanogram of that cDNA to put into our Nextera XC library preparation. The Nextera chemistry utilizes an enzyme for fragmentation and adds adapters through PCR. You can use 10 picograms to 10 nanograms of high-quality total RNA with this workflow. 10 picograms is the typical amount of RNA that can be obtained from one cell, and this kit has been used for single-cell work. You will need 2 kits for this workflow: the Takara Smarter Kit to generate double-stranded cDNA, and the Nextera XC kit for library preparation. Because we're using the Nextera XC workflow for library preparation, we can use the dual indexes from this kit to multiplex up to 384 samples. Keep in mind that this workflow is not stranded. In this matrix, we've compiled key features that are different between the mRNA kits that we just discussed This can be a good reference when you're planning your project. For instance, if strand information is important to you, you may want to consider our stranded mRNA kit. If you have FFPE material, you may consider the TruSeq RNA exome kit. If you only have 50 picograms of input, you will need to use the smarter ultra-low kit with our Nextera XC workflow. Some other factors to consider, it is always important to consult the literature for the best approach for your project. Keep project continuation in mind, so if possible, it would be best to continue using the kit that's already being used for your project. Up-to-date information and details can be found on the Library Prep Kit Selector and support pages linked here. For more information on the Smarter Kit, refer to Takara's website. Now, let's look at whole transcriptome library prep kit options. There are two main kits for this approach, TruSeq Stranded Total RNA and Illumina Stranded Total RNA Ligation. The TruSeq Stranded Total RNA workflow is very similar to the TruSeq Stranded mRNA workflow. The main difference here is that instead of pulling down what we're interested in with poly-T beads, we are pulling out what we don't want. Our RNA can be abundant in a sample, and it doesn't really provide much more information for sequencing. So we deplete our RNA by using sequence-specific probes against our RNA. After our RNA depletion, we do not perform a poly-A step. We proceed with fragmentation, cDNA synthesis that keeps strand information, A-tailing, index adapter ligation, and library amplification. Note here that the fragmentation step can be modified for FFPE samples that are expected to be more fragmented. As I just mentioned, this workflow can use FFPE material as input. And this is because we're removing our RNA, but leaving everything else behind. So even if our RNA is degraded, we will still retain it. We won't see a 3' bias as we would with a poly-A selection. This kit supports depletion with Ribozero, human mouse rat, gold, plant, and globin. Gold has the same probes as human mouse rat, plus probes against mitochondrial rRNA as well. If you're using a sample type that may not be compatible with the Ribozero probes listed here, you have more options. You can use a third-party or a custom rRNA removal kit of your choice to deplete the rRNA up front. Once the rRNA is depleted, you can use the depleted input with either the TruSeq Stranded Total RNA or even the TruSeq Stranded mRNA to proceed with fragmentation, priming, cDNA synthesis, and subsequent library prep steps. The newer Illumina Stranded Total RNA Prep Ligation with Ribozero Plus kit has a very similar workflow to TruSeq Stranded Total RNA. With Illumina Stranded Total RNA, we deplete with an enzymatic depletion method with Ribozero Plus. And at the indexing step, we first ligate index anchors before adding index adapters through PCR. This is in contrast with TruSeq Stranded Total RNA that uses the bead pull-down based rRNA depletion method and directly ligates index adapters. Let's take a closer look at the Ribozero Plus enzymatic depletion and the Ribozero pull-down based depletion. On the left with Ribozero Plus, we use DNA probes that hybridize to rRNA target. RNase H then cleaves the rRNA bound to the DNA probes, and the DNA probes are digested away, leaving us with our rRNA depleted input. With Ribozero on the right, biotinylated probes hybridize to rRNA target. Streptavidin beads are then used to pull down the rRNAs bound to the biotinylated probes, leaving us again with rRNA depleted input. The benefit of enzymatic depletion is that we would expect less rRNA carryover since there are no beads to accidentally pipette into the next step. As with TruSeq Stranded total RNA, strandedness is introduced during second strand synthesis through incorporation of DUTP. Just like the Illumina Stranded mRNA workflow, we use index anchors that introduce a T overhang for ligation. After ligation, we can then add index adapters by PCR. This T overhang can be trimmed using options described in this link. This kit can support as little as 1 nanogram when using high-quality material, or as little as 10 nanograms when using low-quality Win228 material. It provides strand information and can support up to 384 plexity and has around a 3-hour hands-on time. Here we have our matrix for our whole transcriptome kit. The idea behind these kits is to deplete rRNA or other abundant transcripts and then make the library. Once again, information in the literature and project continuation should be considered. If we want to interrogate specific targets, we can utilize one of our targeted kit options. There are enrichment-based options and amplicon-based options. I will not go over the amplicon-based options today as they are covered in other webinars about amplicon sequencing. Enrichment-based workflows tend to be less challenging for larger targets than amplicon workflows, and they can be useful in more exploratory studies as they're not constrained by specific PCR primer design limitations. The enrichment-based targeted RNA kits that we will go over today are Illumina RNA Prep with Enrichment Tagmentation, TruSight RNA Pan-Cancer, TruSight RNA Fusion, and TruSight Tumor 170. We'll begin with the Illumina RNA Prep with Enrichment Tagmentation workflow, which is a tagmentation-based enrichment kit for RNA input. It's essentially the RNA version of the Nextera FlexFor Enrichment Kit, if you're familiar with that. This is now called Illumina DNA Prep with Enrichment. This kit converts RNA to double-stranded tDNA, which can then be tagmented for library preparation. Once libraries are made, probes are used to enrich for the libraries or sequences of interest. Illumina RNA Prep with Enrichment utilizes bead-linked transposomes, specifically EBLTL beads. These transposomes fragment the double-stranded tDNA while adding partial adapter sequences, which are similar to the index anchors used for the Illumina Stranded mRNA and total RNA kits. Index adapters can then bind to these partial adapter sequences during the indexing PCR that will make full libraries. This kit requires 10 nanograms if using high-quality material, and a minimum of 20 nanograms if using low-quality material, depending on the DV200. It accommodates 3-plex spooling for enrichment reactions, and it is compatible with up to 384 unique dual indexes that can accommodate 384-plex sequencing. This workflow has around a 2-hour hands-on time, and the output will depend on the targets of the panel used. Now, the TruSight Pan-Cancer workflow may look familiar as it's the same workflow as RNA exome, where we bind target sequences with biotinylated probes and capture them using streptavidin beads. Instead of probes for the whole transcriptome, we have probes that are targeted against cancer-related RNA transcripts. The input is the same as with RNA exome, but the output is different as we are now targeting cancer-specific transcripts. TruSight RNA Fusion also has the same enrichment workflow, but here we are targeting fusion-specific transcripts. The input is the same, but the output is different. TruSight Tumor 170 is also an enrichment workflow for both DNA and RNA input. It targets 170 genes involved in solid tumor formation, and the RNA workflow covers some of these. Both DNA and RNA can be used as input at 40 nanograms. The 170 genes targeted by this kit can be found within the manifest, which is located on the kit support page. If you're interested in specific genes and would want to know if these genes are part of a probe panel, you can use our Gene Panel Finder tool. With this tool, you can search for your genes of interest and see if there are kits or panels that contain these genes. And you can access this tool through this link. And here is our matrix for our target enrichment sequencing kit options. And altogether, here is our RNA Kit Mega Matrix with the mRNA, whole transcriptome, and enrichment options that we just discussed in this webinar. This can be used to determine which kit is most compatible for your project. Finally, let's talk about some additional resources that are available to you for RNA sequencing. Some resources available on the Illumina website include an RNA sequencing page that talks about all types of RNA sequencing. We also have the Library Prep and Array Kit Selector tool to help select the kit that would be best suited for your needs and input type. The Gene Panel Finder that can help you identify existing panels that contain your genes of interest. A link to the Sequencing Methods Review that contains summaries of some peer-reviewed publications relevant to RNA sequencing. A recorded webinar on small RNA sequencing, introduction and best practices for those of you who are interested in small RNA sequencing. And the links to the product and support pages for the Illumina RNA kits that we just discussed. We have also compiled a list of RNA-Seq related bulletins here for your reference, and at the bottom is a link to all our support bulletins. Thank you so much for taking the time to join me here today. I hope this session has been helpful for you.